Namaskar. Hello and welcome to Know the Nobels Forum 2024. And in this edition of Know the Nobels Forum, we would be covering the personalities and Nobel Prize winning phenomena in the field of chemistry for the year 2024. And this year's Nobel in Chemistry comes with a lot of newness and a lot of differentiated perspectives, whereby this year's Nobel Prize has been given to three Nobel laureates. Uh, One half of the Nobel Prize money has been given to one Nobel laureate, who is David Baker. He has been awarded the Nobel Prize in Chemistry for computational protein design. And the other half of the 11 million Swedish kronos, which is 5.5 Swedish, that will be divided equally between Demis Hassabis and John Jumper for the protein structure prediction. So this year's chemistry Nobel has a lot of imprints from the space of biology, and as we can see, there is a lot of computational aspects overlaid on top of it. So basic chemistry, protein, but then protein exists largely in the biological world, the organic spaces of it all, the biochemistry part of it all. And then there is a lot of computational aspects involved out there. So Nobel is getting increasingly uh, interdisciplinary, multidisciplinary, and Nobel committees are keeping track of the as we can say that the latest advances in technology and things like that. So Nobels are keeping up with the time. One of the oldest and one of the most prestigious prizes, one of the most prestigious platforms, we can say so, because money is not that very important. It's the prestige which lies behind a Nobel. And just for a attraction, like as we've been saying, that Nobel prizes are given for works, foundational works dating back for around 3 decades to 4 decades. In the Know the Nobels Forum, we saw that the work which started on in 1982, uh, it got recognized in 2024. But these two gentlemen, they are in their early 30s, and they have been awarded the Nobel Prize for the first time, and they come from the Google legacy for that matter. So the young Turks, as we can say so, they are now touching upon and computer sciences, as we all know about it. It's not the foundational one. When Nobel was even established, computer was not even invented for that matter. But now the foundational disciplines are getting so very revolutionized by this latest technology or science's latest field on the deck. So as usual, we'll be starting off. We would be taking forward this Know the Nobels forum by depending upon the Nobel prize.org official website. That's where every releases actually come from. And this is the press release. This was announced on 9th of October 2024 by the Swedish Academy of Sciences. The one half of the award goes to David Baker, who is with the University of Washington, Seattle, USA, and Howard Hughes Medical Institute. So, two institutional affiliations. And the other half has been given jointly to Demis Hassabis, Google DeepMind, London, UK. And again, his colleague at the same Google DeepMind, London, UK. So some of the Google's AI work is happening not in America, but in the imperial capital of the world, and London as we all know it. And we saw in the creative earlier that David Baker receives it for computational protein design, and John Jumper and Demis Hassabis they receive collectively for the protein structure prediction. As we will see, there was a lot of application of the AI. So before we come to the phenomena of it all, as we generally do, we want to know, we actually unravel the personalities behind the phenomena, the Nobel Prize winning phenomena. So David Baker was born in 1962 in Seattle, Washington, USA, and his PhD in 1989 from University of California, Berkeley. And he was the professor at University of Washington, Seattle, and investigator at the Howard Hughes Medical Institute. So prize is in chemistry, but the person's affiliation is from the field of medical sciences. So a lot of if we see the physiology award is fine. It went for the microRNA this year. The physics award again went for the. It was on a different part, but then on the computational side, it went for the deep learning, machine learning using physics. But the chemistry award, as we can say, it has a lot of medical implications. And Demis Hassabis, 1976, early 40s. He did his PhD from 2009 from University College London, and he's the CEO of the Google DeepMind London. And John Jumper, much younger chap, 10 years younger than Hessabis. He was born in Little Rock, USA, PhD in 2017. Just imagine the 2017, not even 10 years old PhD. And his PhD was from University of Chicago, and he's a senior research scientist at the Google DeepMind London. So a bit about the Royal Swedish Academy of Sciences. It was founded in 1739, an independent organization whose overall objective is to promote the sciences and strengthen their influence to society. The academy takes a special responsibility for the natural sciences and mathematics, but endeavors to promote the exchange of ideas between various disciplines, and that's what these guys have been doing in. So we'll be straight away going to the popular section, the popular science background, and take the Know the Nobels forum and what the Nobel Committee they say the popular science background. They have revealed this trinity. They have revealed proteins secrets through computing and artificial intelligence. So basic sciences, experimentations, and things like that. We used to know about unraveling the sciences. Now, computational power, which has now come to the mankind, is being used increasingly more and more to unravel the secrets of the universe. And before we advance over here, I would really like to bring in Dr. Surbhi, the president at the Think India Institute. Dr. Surbhi, what is so very important about proteins in the human body? There is a lot of talk, but from a layman's perspective, from a curiosity perspective, you being a eminent medico from All India Institute of Medical Sciences, what is so special about the proteins that it has remained so very, it has, I would say, it has kept the entire scientific community so very puzzled and curious about it.
So, a very warm good evening to all. And, when we talk of proteins, protein is such an important constituent of the human body. When we say the human body, we mean that protein is forming the building block of essential almost at all the levels. We talk of immunity antibodies, they are proteins. We talk of there are glycoproteins on receptors, neuromuscular, cardiac, you name the system. And protein is forming muscle. Protein is forming the basic structure in the body. And all the hormonal systems, the messengers, the mediation of signals in the human body is all being done through protein. So protein is essentially, we can say, the building block. And even when you say talk of genetics, the nuclear material, so the end product of it all is the protein. When translation, DNA to mRNA, mRNA to protein. So what is this protein doing? It is importantly effectuating all the important processes in the human body. So, for example, if you see what we say, very common, I would say, give an example, HbA1c, which is the measurement of the diabetes level in the body. To it's HbA1c. So
Mhm.
that is a glycoprotein. So everywhere it is a biomarkers. It's a hormone. It's a building block. It's forming the muscle. It is in the receptors. It's very, very crucial for the processing and the functioning of the human body.
Thank you.
In
Thank
nutshell.
you, Purvi. I think so. This is where, and before we embark upon a deeper insights into the Nobel Prize in Chemistry for the year 2024, Nobel committee, nobelprize.org hosts the entire range of Nobel Prizes since 1901. And if we just have a glance of it all, in 1901, the first Nobel Prize in Chemistry was given to Jacobus H. Want Hoff in recognition of the extraordinary services he has rendered by the discovery of the laws of chemical dynamics and osmotic pressure in solution. So the entire progress of human understanding and knowledge about the chemical world, perhaps it can be traced back to the Nobel Prizes, the path-breaking discoveries, the path-breaking Nobel Prize winning fundamental discovery of principles, discovery of natural phenomena. It can all be accorded to these Nobel prizes for that matter. I would really like our chemistry faculty to look into it deeply and take this noble narrative back into their classrooms so as to give the students a contemporary view of what lies ahead in interdisciplinarity as we are exploring more and more. They are not physicists of the first order or the chemists by education or for that matter by profession. Now this year, two computer scientists have been awarded the chemistry Nobel Prize because their contribution was out there to solve a chemical problem, which has an implication in the biological field. So let's come through. Let's, in our characteristic fashion, we will read through para by para, sentence by sentence, phrase
In
by.
fact, I would like to come in here, that in medical profession, in the medical curriculum, the first year subject is biochemistry.
Mhm.
That is one of the major subjects. So that is why a synthesis of biology and chemistry, and that is what is biochemistry. So it's so very pertinent and
Lovely,
important.
lovely. Thank you, Dr. Subi. So chemists have long dreamed of fully understanding and mastering the chemical tools of life. Uh, so simple definition of proteins is they are the chemical tools of life. As simple as that. And this comes from no one else than the Royal Swedish Academy of Sciences, which gives the Nobel Prizes, right? So if anyone asks what are proteins, so they are the chemical tools of life. So this dream is now within reach. Demis Hassabis and John Jumper, the twin youngsters, 1976 and 1985 born chaps, they have successfully utilized artificial intelligence to predict the structure of almost all known proteins, like the human genome mapping. They have done uh, to predict the structure of almost all known proteins. This is the kind of expertise they have brought to the humankind. And David Baker has learned how to master life's building blocks and create entirely new proteins. So one is a synthetic, one is like you can predict the structure, you can have an design a blueprint, and David Baker has learned how to master life's building blocks and create entirely new proteins. So combined with they are presenting a revolutionary power to the word per se. The potential of their discovery is thereby so very enormous. So, how is the exuberant chemistry of life possible? The answer to this question is the existence of proteins which can be described as brilliant chemical tools. So now they have put up another adjective. Yeah, they were chemical tools of life, but now they say brilliant chemical tools of life. They are generally built from 20 amino acids that can be combined in endless ways. Now, mathematics comes into play as to how the permutations, combinations can happen. And using the information stored in the DNA as a blueprint, the amino acids are linked together in our cells to form long strings. Then the magic of proteins happen. The string of amino acids twists and folds into a distinct, sometimes unique, three-dimensional structure. Now, the Nobel illustrator uh, uh, has put up in a very childlike fashion. It's for the students, it's for the young learners to fall in love with these Nobel narratives. So they are just playing with some kind of, you can say, two toys or something like that, but they are three-dimensional structures of amino acids. So this structure is what gives proteins their function. So this, like in chemistry 101, we all read about the orientation of molecules, and that's how the properties are given over there. So some become chemical building blocks that can create muscles, horns, or feathers, while other may become hormones or antibodies. So proteins, like for example, when we were there in the Nobel's Forum for Physiology or Medicine for this year, the discovery of microRNAs, the curiosity of the scientists was that how the basic cells, they get differentiated into different functionings. So then microRNA came, that how it is a non-coded messenger RNA, which gives this power, from transcription to translation, the post transcription function which they do, and it gives the various cells their own properties. So similarly, proteins also have various functions, we can say so. So many of the proteins form enzymes which drive life's chemical reactions with astounding precision. Astounding precision. Now, this is what makes human body and the cosmic design so very intricate. Even the most complex of the human-made machines are not so very precise, and maybe the technologists and engineers, they really every day they take inspiration from the biological world, from the real natural world, and we are using artificial intelligence to understand the complexity of the universe. So the proteins that sit on the surface of the cells are also important and function as communication channels between the cells and its surroundings. So again, to reiterate, this is a kind of a pedagogy which we insist at Know the Nobel Forum, that repetition of the foundational terms, repetition of the foundational precepts is very, very, very important, because these are the building blocks, as Dr. Surbi said, of knowledge. Once our once we get initiated into a particular discourse by mastering the fundamental terms, precepts, and have a feel of it all, it comes naturally to us. The knowledge foundation builds up so very fast. So proteins are the chemical tools of life. They are the brilliant chemical tools, and this is what this scientific background says, that it is the structure, the three-dimensional structure into which the string of amino acids twists and folds. That is what gives proteins their function. It's the orientation, like the space is curved. That was the Einstein's discovery, that the time-space complexity, the space is curved. It is not exactly as we see over there. That is why the lunar months, whatever we have got, that's a different physics of it. So spatial spatiality has got a lot of importance in addition to the structure, to the chemical structure. So it is hardly possible to overstate the potential encompassed by life's chemical building blocks. these 20 amino acids. This is what the Nobel Committee, the Royal Swedish Academy scientists, they want to impress upon, that this another dimension of spatiality along with the 20 amino acids results in so much of exorbitant, exuberant chemistry of life. So the Nobel Prize in Chemistry 2024 is about understanding and mastering them at an entirely new level. Now, as we told earlier, one half of the prize goes to Demis Hassabis and John Jumper, the young computational scientists who have utilized artificial intelligence to successfully solve a problem that chemists wrestled with for over 50 years. So, half a century challenge in a jiffy because of the power of AI. So, that's the constructive power of AI, which, if deployed for scientific discoveries, can really work wonders. And the jury is again divided that what will happen if it goes the nefarious way. So predicting the three-dimensional structure of a protein from a sequence of amino acids, this is what they have mastered. This is what they have brought about. So this has allowed them to predict the structure of almost 200 million known proteins. So this is the size, like microRNAs are thousands in numbers, and we say the large language models, the contemporary ones, they have around 1 trillion plus parameters. So 200 million proteins. This is the complexity. This is what they can predict. The other half of the prize is awarded to David Baker, and he has developed computerized methods for achieving what many people believed was impossible. So, chemists, the titration, the beakers. So, chemistry labs have now, in addition to all that chemical compounds and things like that, it has come to the computer simulation part of it all. That's the power of information. And so he creating proteins that did not previously exist, and which in many cases have entirely new functions. So scientists say कि भाई 200 मिलियन प्रोटीन्स हैं, तो क्या हम नए प्रोटीन भी बना सकते हैं? So that's the kind of power which David Baker has shown. So the Nobel Prize in Chemistry recognizes two different discoveries, but as you will see, they are closely linked. And to understand the challenges this year's laureates have overcome, we must look back to the dawn of modern biochemistry. So, this is what Dr. Surbi was talking about, that the first year undergraduate medical program has the subject, the foundational subject of biochemistry. So, the first grainy pictures of proteins, I would like to give the honor of Dr. Surbi to take on this section to explain.
So, chemists have known since the 19th century that proteins are important for life's processes. But it took until the 1950s for chemical tools to be precise enough for researchers to start exploring proteins in more detail. Cambridge researchers John Kendrew and Max Perutz made a groundbreaking discovery when, at the end of the decade, they successfully used a method called X-ray crystallography to present the first three-dimensional models of proteins. In recognition of this discovery, they were awarded the Nobel Prize in Chemistry in 1962. So in this diagram, we can see, the amino acids, various amino acids have been mentioned in the diagrammatic form. We start with lysine.
Color combinations, what the Nobel illustrator has used. I think this should be a chart in every chemistry class for that matter. Please, please.
Absolutely, absolutely. So you can see these various amino acids: lysine, leucine, isoleucine, histidine, phenylalanine, arginine, tyrosine, serine, proline, glycine, glutamic acid, glutamine, cysteine, aspartic acid, asparagine, alanine, valine, tryptophan, threonine, methionine. My God, these terms we have been reading so very frequently during our medical studies, and these are the building blocks. These are the amino acids which form the essential proteins in the body. Some of this is going into enzymes. We have you might have heard of in your blood reports SGOT, SGPT. So all these are enzymes: serum glutamine, phosphotransferase. They're combinations of these amino acids somewhere. For example, you can see lysine. We have tyrosine. all these are something is going in the cerebrospinal fluid, something in the salivary saliva, so many of these are into digestive enzymes, and of course nuclear material, which is forming the proteins. So um, amino acids are the building blocks, and then you can see a protein can consist of everything from tens of amino acids to several thousand. The string of amino acids folds into a three-dimensional structure that is decisive for the protein's function. So this is what they've said. And subsequently, researchers have primarily used X-ray crystallography and often a great deal of effort to successfully produce images of around 200,000 different proteins, which laid the foundation for the Nobel Prize in Chemistry 2024.
So, a riddle, how does a protein, and one more important thing, the purpose, the takeaway message is just to master some of these terms, just to have a basic feel of what this phenomena is all about. We don't expect the young learners or even the Nobel nurturing teachers in chemistry to really master it to its end depth, but to have a macroscopic view, a feel of things, and to be able to link it across with the curriculum which is out there, whatever level we are. I think this is the continuum which we wish to establish, like from a sixth grade, a post primary level of a chemistry student to the post doctoral level of a chemistry scientist. So this kind of we can create a kind of a kind of a connection through these Nobel, know the Nobel forums. I think that will be a success over there. So how does a protein find its unique structure? This has been a riddle, a puzzle for almost 50 years. That is what they have mentioned about. And these two young, as you can see on the backdrop, these two young computer scientists, they have solved this riddle. So Christian Anfisen, an American scientist, made another early discovery. And using various chemical tricks, he managed to make an exciting protein unfold and then fold itself again. So folding is important. And now folding, at times when we come in the senior classes, we don't understand कि जो छोटे बच्चे होते हैं वो पेपर को फोल्ड कैसे लगे रहते हैं. They have that elemental, I would say, the raw intelligence in them. They keep on folding something here and there. So I think this folding thing, we can really go to that pre-primary classes and see how these kids are folding pieces of paper and taking joy over there. And that's where the basic structure of human life, the very intelligent chemical tools of life work. So the interesting observation what Christian Anfisen made was that the protein assumed exactly the same shape every time. So he did some chemical tricks for that matter. In 1961, he concluded that a protein's three-dimensional structure is entirely governed by the sequence of amino acids in the protein. Now, this was a major discovery. The sequence determine its three-dimensional structure. This led to him being awarded the Nobel Prize in Chemistry in 1972. So we can see there was one earlier Nobel Prize as of 1962, which this was one research, John Kendrew and Max Perutz, and the second one was 1972, 10 years hence. So, however, Emphasen's logic contains a paradox, which another American, Cyrus Leventhal, pointed out in 1969. He calculated that even if a protein only consists of 100 amino acids, in theory, the protein can assume at least 10 raised to power 47 different three-dimensional structure. If the chain of amino acids were to fold randomly, it would take longer than the age of the universe to find the correct protein structure. So, in a cell, it just takes a few milliseconds. So, how does the string of amino acids actually fold? So Anfinson's discovery and Levinthal's paradox implied कि कुछ अलग हो रहा है, something else is happening. That folding is a predetermined process. It's a pre-programmed process. somewhere or the other. And that's where AI can be brought into place for pattern recognition for something previously learnt over there. And importantly, all the information about how the protein folds must be present in the amino acid sequence. That information should be there. So between these two discoveries, it came through. So throwing down the gauntlet for the great challenge of biochemistry, the story starts getting interesting. And the above insights led to another decisive realization. If chemists know a protein's amino acid sequence, sequencing is something which they have known early, they should be able to predict the protein's three-dimensional structure. This was an exciting idea. If they succeeded, they would no longer have to use fiddly X-ray crystallography and could save masses of time. They would also be able to generate structures for all the proteins where X-ray crystallography was not applicable. So these logical conclusions threw down the gauntlet for what has become the great challenge of biochemistry, the prediction problem. So to encourage more rapid development in the field in 1994, researchers started a project called Critical Assessment of Protein Structure Prediction, CASP, which developed into a competition. So every other year, researchers from around the globe were given access to sequences of amino acids in proteins whose structures had just been determined. So a repository started building up. However, the structures were kept secret from the participants. Only the sequence were unraveled. It's a kind of a puzzle. So the challenge was to predict the protein structures based on the known amino acid sequences. So CASP attracted many researchers, but solving the prediction problem proved incredibly difficult. The correspondence between the predictions researchers entered into the competition and the actual structures hardly improved at all. So the breakthrough actually occurred in 2018, very uh, recently, when a chess master, neuroscience expert, and a pioneer in artificial intelligence entered the field. So, board game master enters the protein Olympics. So just see the way the Nobel Committee has written this very abstruse, very big challenge in such a readable fashion, such a comprehensible fashion for all of us. So let's take a look at Demis Hassabis' background. He started playing chess at the age of 4 and achieved master level as a 13-year-old. So in his teens, he started a career as a programmer and successful games developer. He began exploring artificial intelligence and took on neurosciences, where he made self revolutionary discoveries. He used what he learned about the brain to develop better neural networks for AI. In 2010, he co founded DeepMind, a company that developed masterful AI models for popular board games. The company was sold to Google in 2014, and 2 years later, DeepMind came to global attention when the company achieved what many then believed to be the holy grail of AI, beating the champion player of one of the world's oldest board games, Go. However, for Hessabis, Go was not the goal. It was the means for developing better AI models. After this victory, his team were ready to tackle problems of greater importance for humanity. So in 2018, he registered for the 13th CASP competition. So it was an open competition, critical assessment of protein structure prediction. An unexpected win for Demis Hassabis AI model. In previous years, the protein structures that researchers predicted for CASP had achieved an accuracy of 40% at best. With their AI model AlphaFold, Hassabis team reached almost 60%, and they won, and the excellent result took many people by surprise. It was unexpected progress, but the solution was still not good enough. For success, the prediction had to have an accuracy of 90% when compared to the target structure. So now how AlphaFold 2 work? It is their AI model. And as part of the AlphaFold 2's development, the AI model has been trained on all the known amino acid sequences and determined protein structures. So these are the databases. So this is a whole illustration. We'll just try to explain as much as possible. So data entry and database searches. An amino acid sequence, the 20 amino acid with unknown structure is fed into AlphaFold 2, which searches databases for similar amino acid sequences and protein structures. So amino acid sequence, AlphaFold 2 is the program, and they try to search from the databases कि इस sequence के साथ कौन सा structure होगा, the three-dimensional folding, how it is folding. So sequence analysis, the AI model aligns all the similar amino acid sequences, often from different species, and investigates which parts have been preserved during the evolution. So unknown sequence have co-evolved across species. In the next step, AlphaFold 2 explores which amino acids could interact with each other in the three-dimensional protein structure. Interacting amino acids co-evolve. If one is charged, the other has the opposite charge. So they are attracted to each other. If one is replaced by a water repellent, which is a hydrophobic amino acid, the other also becomes hydrophobic. So this is the kind of sequence analysis this AlphaFold 2 does, that amino acids in folded protein structures. So charged, plus minus, and hydrophobic. So using this analysis, AlphaFold 2 produces a distance map that estimates how close amino acids are to each other in the structure. How close are they in the structure? So furthest apart and the closest. So then comes the stage of AI analysis. So using an iterative process, AlphaFold 2 refines the sequence analysis and distance map. The AI model uses neural networks called transformers, which have a great capacity to identify important elements to focus on. Data about other protein structures, if they were found in step 1, is also utilized. So cycle 1, cycle 2. So this is how they try to, from a sequence of amino acid, they try to predict the structure. So hypothetical structure they create. AlphaFold 2 puts together a puzzle of all the amino acids and test pathways to produce a hypothetical protein structure. This is rerun through step 3. After 3 cycles, AlphaFold 2 arrives at a particular structure. The AI model calculates the probability that different parts of this structure corresponds to reality. So this is an iterative process through which the exact up to 90% of probability the protein three-dimensional structure is how the amino acids have folded themselves is arrived at. So Hassabis and his team continued developing AlphaFold, but however hard they tried, the algorithm never quite went all the way. The hard truth was that they had come to a dead end. The team was tired, but one relatively new employee had decisive ideas about how the AI model could be improved, and he was John Jumper. And as his surname said, he jumped this algorithm, this AI model, to come to a breakthrough in finding out the exact structure of the protein. So John Jumper picks up the gauntlet of biochemistry's big challenge. So John Jumper's fascination with the universe was what made him start studying physics and mathematics. However, in 2008, when he started working at a company that used supercomputers to simulate proteins and their dynamics, he realized that knowledge of physics could help solve medical problems. In the Know the Nobels Forum on Physics, we saw that how the basic foundational physics was used for machine learning, deep learning, how the Nobel laureates H and H, they showed it across through Boltzmann machine and so on and so forth. So Jumper took this newly acquired interest in proteins with him when in 2011 he began his doctorate in theoretical physics. To save computer capacity, something that was in short supply at the university, he started developing simpler and more ingenious methods for simulating protein dynamics. Soon he too picked up the gauntlet of biochemistry's big challenge. In 2017, he had recently completed his doctorate when he heard rumors that Google DeepMind had, in great secrecy, started to predict protein structure. So, again, these fundamental challenges of sciences, they are kept as some kind of state secret, very secret, because this is the power. If it is unraveled over there, it can really change the face of humanity. He sent them a job application. His experience of protein simulation meant he had creative ideas about how to improve AlphaFold, which is the AI model at Google DeepMind. So after the team had started to tread water, he was promoted. So Jumper and Hassabis co-led the work that fundamentally reformed the AI model. So astounding results were achieved with a reformed AI model. The new version, AlphaFold 2, was colored by Jumper's knowledge of proteins. So Jumper came with a deep knowledge of proteins. Uh, 20 million of them we have just read. That's the number of proteins which have been mapped so far. But the structure prediction is the challenge. The team also started to use the innovation behind the recent enormous breakthroughs in AI, neural networks called transformers. So this is a most advanced form of neural networks. The uh, uh, these can find patterns in enormous amounts of data in a more flexible manner than previously and efficiently determine what should be focused on to achieve a particular goal. So the team trained AlphaFold 2 on the vast information in the databases of all known protein structures and amino acid sequences. So AI models, mota, we all have an idea, but then what differentiation is made that makes all the difference in a particular AI model. So the team trained Alpha 2 on the vast information in the databases of all known protein structures and amino acid sequences. And the new AI architecture uh, uh, started delivering good results in time for the 14th CASP competition. It started in 1994. So in 2020, when CAPS organizers evaluated the results, they understood that biochemistry's 50-year-old challenge was over. It was over. It was championed. In most cases, Alpha Fold 2 performed almost as well as X-ray crystallography, which was astounding. So, as chemists, as chemistry faculty, we all understand that X-ray crystallography is an exact probe using the X-rays to determine the protein structure, but computationally, and to do it for the uh, group, entire group of proteins is so very challenging. So when one of CASP's founders, John Moult, concluded the competition on 4th December 2020, he asked, what now? What now? Mankind's challenge of determining the protein structures comprising of 20 amino acids folding in multiple ways. So we will return to that. What now? Now we are going to go back in time and shine a light on another participant in CASP. Let's present the other half of the Nobel Prize in Chemistry 2024, which dealt with the art of creating new proteins from scratches. So it is not an arbitrary combination of amino acids in space which creates a protein. There is an art and craft of it, which David Baker, a textbook about the cell, makes David Baker change direction. So when David Baker started studying, so for all our Nobel nurturing teachers, this is so very important that the story behind a Nobel phenomena is the core objective of these Know the Nobel forums, and that's how the Nobel narrative can enter the classrooms to enthuse, to excite the young learners that these are the frontiers of academic learning, and it's more suspenseful than even a detective serial for that matter. So when David Baker started studying at Harvard University, he chose philosophy and social science. So that is where NEP 2020 focuses a lot on interdisciplinary learning. UGC, for that matter, under the stellar guidance of Professor Jagdish Kumar. Now, students can opt for any subject combination for that matter. And just see the Nobel Prize winner of chemistry, David Baker, he chose philosophy and social sciences, and eventually he bagged the Nobel Prize in chemistry. So let's see the mystery, let's see the story. However, during a course in evolutionary biology, he came across the first edition of now classic textbook, Molecular Biology of the Cell. I think I would like to invite Dr. Surbhi to tell more about this classic book, Molecular Biology of the Cell, and then we will go forward.
So, before I start, I would like to really welcome my biology teacher, Sir C.S. Mishra, sir. He's there with us on the forum, who has taught me biology and who was the driving force behind my becoming a doctor. Welcoming you, sir, on the forum. Request you to kindly come on camera and if you can speak a few words. And my guru is here. I would like to just invite sir to speak a few words, sir. Sir C.S. Mishra, sir. Pranam, sir. So you need to unmute yourself, sir.
Yeah, I'm unmuted.
Sir, we can
How
hear
are
you,
you,
sir.
Sudeep? How are you?
Very fine, sir. Your blessing, sir.
Uh, just I because I've just made myself free for this session, and
Sir,
as you requested that please visit, and I'm finding that the it's very enlightening whatever
sir.
sir is telling us, and it will help ultimately the people if they take the full interest to understand what has happened, how the Nobel laureates have worked. So all
True, sir.
I'm very glad to see your work, and I'm always in connect with you. Whenever you send any message, I see it very thoroughly. I thought about 3 or 4 days back only when you invited me for this to send a message, but because of busy schedule, I was not able to do that. So I'm thankful, and I'm part of the session. Don't worry.
Thank you, sir.
Okay.
Thank you for your benediction,
Thank
sir.
you. Thank you. Thank you.
So, so molecular biology, when we talk of, molecular biology of the cell, so this is such an important topic. I mean, what to say when my guru is there right in front of me. So basically, David Baker, when he started studying um, evolution and cell is the smallest unit of life. We know that. And how molecular biology is so very enthralling, it talks from the nucleus to the cell constituents and the DNA. This is where it is the fundamental, I should say, the unit of biology. So cell molecular biology is today the gung-ho word, the buzzword, and this is where I think we can take our study further.
So, from a student of philosophy and social science, he was doing a course on. That's why interdisciplinarity is so very important. कि कभी पता नहीं कि किसका passion और fascination किस field में जाग जाए. So that's why multidisciplinarity, चखना सभी विषयों को, बिकॉज दर्शन और उसका जो सामाजिक रूपक है वो कहीं ना कहीं से उत्प्रेरित हो जाता है. दैट्स व्हाट वी इन इंडिया आर नाउ ट्राइंग आउट इन आवर schools and universities for that matter. So when he went through the textbook of molecular biology of the cell, this led to him changing his direction of life. He began to explore cell biology, and eventually he became fascinated by protein structures. So DNA is one part of it, but at the fundamental level, the protein structures, as we saw that proteins are the chemical tools of life. They are the brilliant chemical tools of life, uh, guided by the DNA for that matter. And when in 1993 he started as a group leader at the University of Washington in Seattle, he took on biochemistry's great challenge. So, using clever experiments, he began to explore how proteins fold, घूमते कैसे हैं, how they fold, and this provided insights he took with him when, at the end of 1990s, he began to develop computer software that could predict protein structures, which was Rosetta. So that was a different paradigm. AI is a different paradigm altogether, and it is progressing phenomenally at a whatever we can say, at a breakneck speed, we can say so. So Baker.
And we can see the juxtaposition of the multidisciplinaries again here. So, wherein we see that molecular cell biology, physics, chemistry, AI, everything is getting juxtaposed.
Yes. So Baker made his debut at the CASP competition. So the common thread across is the CASP competition. And again, for the sake of memory, CASP competition, as they talked about, CASP competition is critical assessment of protein structure prediction. The CASP competition, he also became a contestant. Yes. So in 1998, just 4 years after the CASP was announced, and using Rosetta, and in comparison to other participants, he did really well. So this success led to a new idea that David Baker's team could use the software in reverse. इसको उल्टा use करो. Like we use the reverse osmosis for cleaning the water, the phenomena of osmosis for cleaning the water, getting the minerals ousted, and getting pure drinking water. So instead of entering amino acid sequences in Rosetta and getting protein structures out, they should be able to enter a desired protein structure and obtain suggestions for its amino acid sequence. So, which would allow them to create entirely new protein. So, it's a kind of a recipe in the kitchen कि आज कुछ नया खाना है, बेसिक इंग्रेडिएंट्स रिमेन द सेम, बट आज कुछ इनोवेट किया जाए, आज कुछ फैसिनेटेड न्यू चीजें बनाई जाए. सो एट द एलिमेंटल लेवल ऑफ लाइफ, I think David Baker, perhaps his inroads into social science and philosophy, because science, all the PhDs, they are doctorate of philosophies. So you philosophizing, भारतीय ज्ञान परंपरा में भी दर्शन, विज्ञान और व्यवहार, ये तीन का एक त्रयी की मान्यता है कि there has to be a foundational philosophy for anything, whatever you do, and then there is a विज्ञान of it all, and then at the end of the day, the व्यवहार, the application part of it all. So baker becomes, and name baker. We know bakers, bakery, you're baking. The dough is the same, the fermented dough, the various ingredients, and you bake new delicacies over there. So baker was baking new proteins for that matter. And he became a protein constructor. So the field of protein design, where researchers create bespoke proteins, bespoke tailors we have, कि कपड़े का थान है और उसको आपकी फिटिंग के अनुसार bespoke tailors. So bespoke proteins with new functions began to take off at the end of 1990s, because as we have understood earlier, it is the structure of the proteins which give its all its functions. That is the secret of the chemical tools of life. In many cases, researchers tweaked existing proteins so they could do things like breaking down hazardous substances or functioning as tools in the chemical manufacturing industry. However, the range of natural proteins is limited. To increase the potential for obtaining proteins with entirely new functions, Baker's research group wanted to create them from scratch. And as Baker said, if you want to build an airplane, you don't start by modifying a bird. Instead, you understand the first principles of aerodynamics and build flying machines from those principles. So, a unique protein sees the light of the day. This is figure 3, top 7. The first protein that was entirely different to all known existing proteins. So, a kind of a synthetic protein, we can say so. The name is top 7. The field in which entirely new proteins are constructed is called de novo design. The research group drew a protein with an entirely new structure and then had Rosetta compute which type of amino acid sequence could result in this desired protein. To do this, Rosetta searched a database of all known protein structures and looked for short fragments of proteins that had similarities with the desired structure. Using fundamental knowledge of proteins' energy landscape, Rosetta then optimized these fragments and proposed an amino acid sequence. So, to investigate how successful the software was, Baker's research group introduced the gene for the proposed amino acid sequence in bacteria that produced the desired protein. So one was the computational design, जैसे कि architect does a blueprint and algorithm is designed, but then the actual synthesis, that is what Baker's research group also did. He was a chemist after all. So research group introduced the gene for the proposed amino acid sequence in bacteria that produced the desired protein. Then they determined the protein structure using X-ray crystallography. It turned out that Rosetta really could construct proteins. The protein that the researchers developed, which was named as Top 7, had almost exactly the structure they had designed. So, at such a molecular level, such a cellular level, constructing a de novo protein was truly a path breaking discovery. So, spectacular creations from Baker's laboratory. Top 7 was a bolt from the blue for the researchers working on protein design. Those who had previously created de novo proteins had only been able to imitate existing structures. Top 7's unique structure did not exist in nature. Also, with its 93 amino acids, the protein was larger than anything previously produced using de novo design. So Baker published his discovery in 2003, 20 years back. This was the first step in something that could only be described as an extraordinary development. A few of the many spectacular proteins created in Baker's laboratory can be seen in figure 4. Let's have a look. This is 2016 new nanomaterials where up to 120 proteins spontaneously linked together. 2017 proteins that bind to an opioid called fentanyl, the purple. These could be used to detect fentanyl in the environment. 2021 nanoparticles, which are yellow, with proteins imitating influenza virus on the surface, which is green, and that can be used as a vaccine for influenza, successful in animal models. And 2022 proteins that function as a type of molecular rotor. So these are the building blocks, the chemical tools, and geometrically shaped proteins that can change their shape due to external influences. They can be used for producing tiny sensors. So he also released the code for Rosetta. So a global research community has continued to develop the software, finding new areas of application. So it is time to tie up the loose ends of Nobel Prize in chemistry. What now? That was a question when the CASP problem was solved, and the date we all remember, 4th of December 2020. It was the height, almost close of the COVID pandemic, we can say so. What now? John Malt posed this question, what now? Let's come to that. Just a moment here. So it's like furthering the envelope of scientific quest, scientific curiosity over there. Let's move on. Yes, yes, yes. A lot of it would have gone through our heads, no doubt about it. And that is the whole purpose of it. We are just soaking these very deep Nobel precepts. And I think a couple of iterations of reading would start illuminating the real feel of it all. So, dizzying development for the benefit of humankind. As Nobel Prize Sir Alfred Nobel, in his will, in his famous will, said, the greatest benefit to the mankind. These awards have to go. So, proteins' amazing versatility as chemical tools is reflected in the vast diversity of life that we can now so easily visualize the structure of these smaller molecular machines is mind boggling. It allows us to better understand how life functions, including why some diseases develop, how antibiotic resistance occurs, or why some microbes can decompose plastic. The ability to create proteins that are loaded with new functions is just as astounding. This can lead to new nanomaterials, targeted pharmaceuticals, more rapid developments of vaccines. COVID-19 vaccine was developed in a record time frame because of these advances across scientific disciplines. Minimal sensors and a greener chemical industry, to name just a few applications that are for the greatest benefit of mankind. So, friends, this was all for the Nobel Prize in Chemistry, David Baker for computational protein design, and Demis Hassabis and John Jumper for the protein structure prediction, and very young people how they used AI to really, I would say, predict the protein structure, the 50-year-old problem of biochemists. It was all solved in a jiffy. So this here we come to the end of it. Know the Nobel's Forum for Chemistry, and how chemistry is biochemistry, chemistry juxtaposed to that, solving the mystery of life. I think in this fourth industrial revolution, the convergence of technology, I think the bigger focus is actually going to understand life, how human bodies function, how the yeah, Chaitanya Prakriti, how it functions, and that's where the entire convergence is all happening. Uh, I would request Dr. Surbhi for her closing words, and we would be back for next Know the Nobels Forum, which will be a juxtaposition of Nobel Prize in Peace and Literature. So literature is the human expression. Again, the large language models of AI, they have a lot of language part, because natural language is the OS, it is the operating system of the human cerebrums. And peace is again the social sciences. So after the three fundamental scientific disciplines of physiology, physics, and chemistry, they are so very beautifully intertwined. We will be moving to the next three, which have been clubbed in two Nobel Nobel sessions, uh, Friday and Saturday. So last would be on economics, a very interesting concept in how human psychology works at the emotional level, how the human responses happen. So Nobel has become quite a double helical structure: the fundamental sciences and then the social sciences, peace, literature, which is the poetical expression, what it should be, expressing the human aspirations, human desires. And economics, obviously, the word is all economically so very deeply connected. Dr. Surbi for the closing words, and we will move on. Yeah.
So I take this opportunity to, thank all our listeners, and we are joined by Deepti Ambulkar ji. She's Deepti ma'am. You are from Edify School, Bengaluru, also. Yeah. So
Yes, ma'am. Good evening,
Okay.
ma'am. Good
Good
evening.
evening. Good evening.
Yeah, I'm from Edify School, Bengaluru. Uh,
Bengaluru.
yes,
The
ma'am.
chemistry faculty.
Actually, biology, chemistry, both the
Okay,
subjects I'm
okay.
taking.
Yes, yes.
Yeah.
So biology and chemistry are intertwined. So this is a great. This has been a very great and enlightening session. Thank you, Raghav, for such an in-depth explanation about the protein structure prediction, computational protein design, and we can see that how going from 0 to 1 and then back from 1 to 0. So creating the protein in de novo, which is the synthesizing the protein and then cracking the protein structure, both have been so fundamental to the such wide applications we studied, and in understanding the human functioning, antibodies, vaccines, sensors, and so on and so forth. So this is a clarion call to all our educators to take this to the classrooms pedagogy and scintillate the human minds, the students, to go that extra mile, go the outer box, and go the interdisciplinarity way. Thank you so much once again from all of us at Rethink India, and taking this Nobel pledge, inviting the Nobel nurturing teachers to join in. And of course, we have felicitations in all these disciplines. And thank you so much once again. And
Thank you, Dr. Surbi. We will be resuming this for the next two sessions as well.